Okay. And you know what? We're one past, but I think folks can just trickle in. And Robert, I'm going to turn it over to you if you're ready to go, if you want to share your screen.
Afternoon, and thank you, Michelle. Let's see here. Assuming you can all see that.
Yes, we're seeing all the slides. You're good now.
All right, good stuff. Always nice when everything works. So,
Yes.
once again, my name is Robert Soames. I'm with NJDEP Fish and Wildlife Endangered Non-Game Species Program. Among the many things I do for non-game is freshwater mussel conservation. And I'm a Pinelands regional biologist, handle like management planning and conservation planning in that region. Then I handle our invertebrate conservation work related to insects and then freshwater mussels. We also have a new staff member, Alex, who's I think on the call today as well, who is also learning freshwater mussels and going to be taking on some of our aquatic stuff as well in the future. So I'm going to give like an overview of freshwater mussel conservation, a little bit about like biology, life history, and then how the dams impact them, and then some of the regulatory side of things. And I'm going to try, I have a couple of videos that are embedded in this. I'll try and maybe leave those to the end. I mean, they are really interesting, but I want to make sure we have plenty of time for discussion at the end and we go over to the key points. And I also do like an in-person kind of workshop on freshwater mussel identification, where we do like a lot of hands-on looking at shells and identifying mussels. So like kind of go over some of the features and things related to that quickly, but then some of it's more conducive to actually holding the specimens in your hands. So let's see if we can get this. Switching slides here. All right, there we go. Freshwater mussels are like a vital ecosystem component, part of the food chain, for a lot of different animals. They're water quality indicators, especially a lot of the rare species are very sensitive to water quality changes. Another interesting thing, a lot of people like look into their value for cleaning water. They're naturally, filter feeders, and they vacuum the water, and a lot of people look at it as a positive thing. And an interesting kind of note to think about that as well is not just cleaning the water, but it's also fixing like carbon, nutrients, and materials out of the water into the substrate. So this organism's filtering like free floating organic materials and, algae and other things like that. And then, they extract nutrients from it, and then they discharge it as waste. But that waste is now kind of concentrated and heavier and sinks to the bottom and settles into the substrate of the stream. So they actually fix like carbon in the environment. So it's another like interesting part of how mussels work. They're one of the most rapidly declining species groups in North America, unfortunately. They're negatively impacted by so many different things that everybody knows that impact our waterways. So it's not a surprise that this is one of the species groups that are declining the most. Basic taxonomy, bivalves. We only have one family in New Jersey. So in worldwide, there's 6 families, 165 genera. And in North America, it's 281 species. An interesting note is the majority of the global biodiversity for freshwater mussels occurs in North America. The vast majority of species are found in North America. Like the biodiversity of freshwater mussels in the rest of the world is rather limited. A lot fewer species. There's far greater numbers of species in North America. And then even in New Jersey, our species list is relatively limited compared to like you get out in the Mississippi drainage and things, where there's over 100 species found in that river system. New Jersey, we're limited to 12 native species and then 3 introduced native species. And then 1 species that's been historic and extirpated. So, of these species, the majority of them have a listed status in one way, shape, or form, because the vast majority of them are suffering population declines. And then, like so many people see among so many species, there's less and less common birds, there's less and less bees. The same thing happens with some of the common mussels. Like our common species, like the Eastern Elliptio, are even like declining, and there's less of them than there used to be. So dwarf wedge mussels are federally endangered listed species. And then our 2 endangered species are green floater and brook floater, with green floater, likely to have been extirpated from New Jersey. There hasn't been a record since probably the early 90s now. Our threatened species are triangle floater, eastern lamp mussel, yellow lamp mussel, tidewater mug, and eastern pond mussel. And then creepers are one special concern species. And then when we go into like identification, and when we do the identification workshop, eastern floater, ally floater, eastern elliptio. It's like the focus is to learn the common species. Once you if you want to learn more about mussels, learn about identifying mussels, focus on learning the 3 common species, because if you can identify those and rule those out, you now can kind of go to the next step of the decision tree of which rare or species is it. And then we've got 3 introduced natives: the paper pond shell, the lilliput, and eastern pearl, and the giant floater, And then eastern pearl shells are one historic species that was extirpated at least 100+ years ago. It used to be found in the Hudson River and the Delaware River, and very closely linked to salmon, like Atlantic salmon. So with the loss of the host fish, Atlantic salmon, along with the mussel. Giant floater being one of the more recently identified and confirmed introduced natives. So we don't really know much about that species other than the fact that we do know we have it. And we need to do more research on it. Biology and anatomy, they're filter feeders, graze on algae and bacteria and environment, makes them very susceptible to like water pollution, salt, temperatures, anything like that. The shell is composed of 3 layers: the epidermis, middle, and the nacre. The nacre is like the inside shell, like the mother pearl, sometimes different colors. The beak is dorsally raised, inflated area inside the shell. And like, I go through these features, some of these details are not as valuable online, but then when you start to look at shells and I learn to identify them in person, they become valuable. The shell is held together by lateral and pseudocardinal teeth. Is where some of the muscle of the shell is held together by the internal structure of the mussel. Has a large muscular foot, which is used in locomotion. It allows mussels to be able to bury themselves into the substrate, and it allows them to move if they've been dislodged. And then some species of mussels tend to be more mobile than others. So like brook floaters, for example, are very common. It's very often to find them like on the surface with their foot out moving, So some species are more mobile than others and spend more time moving around their habitats. Now, granted, they don't move very far, but there have been quite a few studies where they do like intensive monitoring of tagged mussel populations, and you can have individuals moving up to 50 meters in the course of a season. And that's like across like the topography at the bottom of the stream, 50 meters from where they started at the beginning of summer to the end. But they also move up and down vertically in the substrates. In that, like when it's cold and come to winter, a lot of them bury down in to be insulated from the winter cold water temperatures. And we've had like things like yellow ant mussels and other species that'll be buried up to 18 inches into the substrates. They can bury pretty deeply when they want to. And it does present challenges for surveying when you have a certain number of individuals that are buried out of sight. All the shell characteristics are really important for taxonomy and identification. Just a quick image, so you know the different parts of a mussel. The anterior is like the front where the muscular foot is retracted most of the time. And when you pick it up, they usually pull it in. But the foot allows them to bury down into the substrates. Usually they're kind of this end down into the substrate with the posterior end with the siphons sticking out for them to actively filter. Sometimes they might be slanted or on their side, or this side maybe down a little bit more, but give or take, they usually are buried in so their siphons can be exposed to filter water. And that's one of the features you look for when doing surveys and things like that, is looking for the siphons and the exposed kind of shell of the mussel. And if you get out in the field to survey for these things, like they blend in incredibly well. But like so many things, you get like a search image and you'll start to learn to see the dark line of the shell, or you start to notice the siphons, because they can be completely covered with algae and other things like that, and silt and other materials. But then all of a sudden, like, magic, once you start to learn to spot those kind of features, you'll be able to see the mussels buried in the substrate, even though they might be very well hidden. Just quick looks at the inside of the shell, which, will definitely like keep the group in the loops. I'd like to run another hands-on kind of class again this winter. When you look at the inside of the shell, some important things for identifying the mussels are the presence and number of lateral or pseudocardinal teeth, which are different structures on the shell. And I say the lack thereof, in that some species have no teeth on the inside of the shell, and it helps you identify them. So like if you're seeing mussels or have shells, that you want to identify, taking photographs of both sides and the interior of the shells, if you have just a set of valves, are really important, because you can look at some of those structures and those shapes and be able to identify the mussel from them. Life cycle. This is the most fascinating part of freshwater mussels, is their life history. Is the vast majority of species are closely linked to fish and host fish, and many are specialists to only certain species of host fish. So for a mussel to complete its life history, You have both male and female mussels in the environment. Now, there's so much diversity and so much variation in like everything we study that the same holds true for mussels. There's a certain percentage of they're finding more and more of this, the more research they do on mussels, that in any given population, there's a certain percentage of hermaphroditic individuals that are both male and female and able to self-reproduce. And some species of mussels, that's the majority of them. And it's probably an adaptation to having like isolated populations and, low genetic exchange and things like that over time. But as a whole, you have male and female mussels, and the baby juvenile mussels are called Gokidia. And they are a tiny grain of sand looking small mussel with a hinge and two little teeth on it. And it serves as kind of like a trap door. And what the idea is it attaches to the gills of a fish, or sometimes the fin, or even the skin and things like that. And it clamps onto the gill, and it rides along, and it extracts a certain amount of nutrients in a very small amount. And they also filter. Now, the mussel rides on the fish for several weeks of its life until it metamorphosizes to like a little bit bigger size, and then it drops off. So this is where like a lot of important things kind of come in, and this part of his life history is real important to connectivity, dams, and invasive species. And it's also very important to just see much important, very important to like the specialists and that are entirely dependent on certain species of fish to get them to the right habitats. So like generalist mussels are generally found in ponds and lakes, and they're not very host specific. They can attach to a whole variety of different types of fish, and they're not super sensitive on the habitat type. So wherever they drop off is fine. Now, some of the rarer ones are very closely linked to things like darters and mad toms and other small bottom of the stream fish that are only found in certain types of habitat. So when it comes to like darters, if you're a mussel, you need to get on a darter to get to the right type of habitat, because the mussels are in the same sandy substrates as the darters, and you're depending on that darter to get your offspring to another patch of similar sandy substrate habitat. So if you attach to like a bluegill or a bass or some other type of fish, and you go along for a ride, you might just drop off in a weed bed or an impoundment or someplace else that is not suitable for you and will not survive. So a lot of the rare and specialist ones are dependent completely on certain types of fish. And the key is having that type of fish be able to get your offspring to the right type of habitat. And if you have dams or some other kind of feature that's breaking up the habitat, those fish cannot disperse to get you where you need to go. Now, on the flip side, this is how invasive or non-native mussels move around very easily with fish stocking. Like there's a huge history of fish being stocked all over related to freshwater fisheries, maintaining fisheries, adding different types of species. And what happens is like if you have freshwater mussels in your pond or lake where you rear your fish, you get mussels going along for the ride and turning up in other places. So that's how a lot of our common pond species, particularly like eastern floaters, get moved around. And then that's how a lot of the introduced natives get moved around as well. We may have gotten fish from someplace in the Mississippi drainage, 100 years ago that brought like lilliput or paper pond shell with them along the way. And that is how Chinese pond mussel got here as well, and carp that were imported from China. So it's really important to think about like the life history of mussels and how they move around the environment and how these different things are important. I'm going to skip our videos to the end. They are really good, but I want to make sure we have time. We can always look at it at the end if we have time. They're both on YouTube, and they go into a lot of the details and some nice imagery of how mussels work. The other interesting thing when it comes to being linked to fish is how do you get your Gokidia onto fish? Some mussels depend on just random happenstance where they just release their young into the environment. And if they get attached onto the fish, so be it. If it doesn't, they just release enough of them out there that things will work out. A lot of species of mussels have developed really complex gill lures. And what they are is, like this shape on the tip of the gill of the mussel that looks like a shiner or a darter or a crayfish or any other thing that a fish might be interested in eating. And the idea being that it actually attracts the fish to the mussel. And when the fish goes to try and bite or eat the supposed lure, the mussel will inject all the Gokidia into the fish and have a better chance of them getting attached onto the host fish. And some of these mussels even take this to like a whole nother extreme, in that some of them will release like packets of Glochidia on a little tendril that'll look like a nymph in the stream. There are some species that release their Glochidia in little balls that look like a fish egg in the stream. Like there's a wide variety of adaptations for getting fish to get Glochidia onto them. This is an example that looks like a crayfish. And there's several species like snuffbox mussels. None of these live in New Jersey, unfortunately, but they're in other states. And the actual shell shape has a bit of a cup on the edge of the shell like this. So it's kind of a cup. So the shell kind of opens a little bit. And I'm hoping with the camera and the projection you can see me. It's a lot easier in person. But it has, the shell has a cup shape on it with a little lure inside of it. And what it does is it attracts the fish to the lure. And when the fish gets it, the shell closes and holds onto the fish and then releases Glochidia into the fish. And you can see in this photo of the mussel actually holding onto the fish. And these are all amazing adaptations, considering, like, mussels don't have eyes. They can't see themselves. It's an amazing example of evolution over time, to link fish to mussels and how important they are to each other. We'll save videos for the end, lifespan. They are some of them among the oldest living organisms on Earth. Some individuals can live up to 100+ years. A lot of shellfish can live a very long time. But when it comes to freshwater mussels, some have relatively short lifespans of 10 to 12 years. And, a lot of these streams and waterways are rocky, high flow, flash floods, they have to be able to grow quickly and reproduce quickly, just because they may be suffering mortality because of flooding and getting smashed against rocks and other things like that. So like the different species in the different environments have different adaptations, so some of the rarest species are relatively short-lived just because of the nature of the environments they live in. They have to reproduce quickly. Problem is, declines may not be immediately detectable because of longevity, and they can be functionally extinct. We do run into populations. I've seen elliptios in several streams and rivers where they're all big, old, ancient elliptios, and there's just no reproduction going on. There's no small, younger individuals. And then on the flip side, like with any like short-lived species, like insects, inverts, there's a whole lot of them that fall in this category, is that it only takes a couple bad years of reproduction and then you go extinct. They don't have the time to kind of wait out poor conditions or changes in their environment. Habitat is highly variable. Rivers, streams, lakes, and ponds. Like any group of species, you have generalists and specialists. Some are linked to tidal freshwater. Some are linked to lakes and ponds. Some are only found in like the larger, slower rivers, while others are dependent on clear, cold, sandy bottom streams. One of the biggest things limiting in New Jersey is pH. There are no freshwater mussels in like the Pinelands region, because the higher acidity just, dissolves the shells. They cannot survive in those environments. It is interesting, though, where we have fish stocking on like periphery areas of the Pinelands, you can get some little populations established of common things like eastern floaters. It's like they don't reproduce, but they can kind of get a toehold, so it's always interesting when you see those things that are on the edge of their suitable range. Distribution is, highly limited by host fish and Glochidia. Dams are a big impediment to that, like connectivity, like a big thing, especially for these rare ones that are dependent on small little fish, like a darter. Like a darter's not going through big migrations like a herring or a shad. They're not able to really get through large barriers or other things like that. Their spread into new habitat is relatively slow. And the thing to think with mussels too is like the time scale necessary for some of these changes can be decades to 50, 100 years. Like how long does it take for, a dam to be removed and for fish and mussels to be able to get to where they need to go in that drainage system? Some are limited by like the drainage area, like, how many metapopulations are there? Like some are just associated with tidewater. So like of our species, you have tidewater mucket and eastern pond mussel that are both pretty much like South Jersey mussels, I call them, because they're pretty much from Trenton south into Delaware River drainage. Calcium also limiting because they need it for shell development. Some of the biggest threats, like I said, the most rapidly declining animal group. Most of the decline's been due to habitat degradation, like so many things we see in our rivers and waterways. Hydrology changes, siltation, runoff, temperature changes, water chemistry changes, pollution, all the standard kind of things. But some of the things that like people don't think about first, everybody always thinks about stereotypical pollution, But on the flip side for mussels, it's just the flow changes make a big difference because they're really dependent on stability of their habitats where they can have places to like be suitable substrates that's stable and not getting washed away, and they're not getting bashed to death on other rocks and things. And what happens, especially in New Jersey, you see a lot of places, is that like the stream flows are entirely changed. Like the water may be pretty clean, but there's just so much runoff and so much flashiness and so much changes to the hydrology that is, flushing out the suitable substrates. It's just constantly washing the mussels and dislodging them, that over time they suffer attrition. Temperatures, climate change, water temperatures getting higher, like some of these species are going to hit some kind of temperature limits, either for their host fish or for them. Invasion of exotic species. We have the Chinese pond mussel in New Jersey, that could displace our native mussels, because invasive species compete for space and food resources. All your stream alteration stuff. So dams being the most obvious thing, but then like channelization, a lot of streams and rivers were straightened over time, bends were removed, and what happens when you have like a natural stream with bends and structures and log jams and all this other stuff, you end up with like a huge mosaic of different substrates on the bottom where mussels can find their home and fish can find their home. When it's kind of become channelized and cleared out, like everything flushes out, like it just kind of clears any kind of diversity to the stream bottom. You can have like just shifting stream bottom changes due to hydrology, in that like your flow rates have now changed so much that any of the substrates that may have been historically stable are now just moving down the stream, just moving along because there's just too much flow. Dams, they alter the physical, chemical, and biological stream environment, increased temperatures, changes in the flow rates. They can provide like, food resources to mussels because, there's a lot of organic material builds up in a pond, but like the overall negative kind of impact is pretty significant in that like your larger dams, pretty much become like, no man's land of mussels. You don't really find anything, or you might find some floaters in there and things like that. Also particularly important is the loss of the host fish. You have things like darters and other things on the bottom of the stream that need cold, clear, sandy bottom water, and now you've got a pond, that's full of lake, bass and sunfish. It's just, the host fish are gone and the connectivity is gone. It gets kind of complex in that dams also can create habitat, but it all depends on the size of the dam and what it's like. And we'll go into some photos, some examples to kind of get into that as well, because like, sometimes you get the small tumbling, low head dams where they kind of create a break-in habitat, and they also create some substrate buildup, so it always becomes like a challenge in that, like, well, some dams are bad, some are like not really doing any harm, it's always complex. Contaminants, heavy metals, pesticides, sewage treatment plants, effluents like ammonia. Mussels are incredibly sensitive to ammonia. Road salt runoff. Other things interesting is copper. They're really sensitive to aquatic herbicides. Salts and copper, they're really sensitive to copper salts and things like that. And the challenge a lot of those chemicals is they're pretty regularly tested on like fish and insects for mortality, but there's been not as many studies on freshwater mussels. And what the studies that have been done show mussels to be about 50 times more sensitive to these herbicides and chemicals than any other organism. So like you might have something that's not causing a fish kill, but it will kill off the mussels in a pond. Continuing on our threats discussion, our non-indigenous species, the Asian clams, which everyone sees everywhere. They've been established in North America for a very long time. They're pretty much ubiquitous. It's hard to say what negative impacts they've had because there's been so many cumulative impacts on freshwater mussels. But in areas where calcium is limiting, sometimes you have substrates in the bottom of the stream that is like a majority of Asian clamshells, and there's an awful lot of calcium being fixed and pulled out of the environment. Zebra and quagga mussels, we don't have in New Jersey. Let's keep it that way. But they cause a lot of problems for mussels and other places in that they just completely encrust and cover over the mussel. Chinese pond mussel, first North American occurrence was in New Jersey, unfortunately. And they are causing a huge problem in Europe right now. And then sea level rise, especially like for our southern species. Tidewater mucket and eastern pond mussel are only found in the lower Delaware River. Like they're going to like hit a wall around the fall line in Trenton at some point for moving their distribution northwards. Like they're going to run out of habitat and they're going to run into like barriers to just naturally migrate northward as the sea level rises. Chinese pond mussel, just a quick touch on that, was in Hunterdon County in an old fish farm that had imported carp 50 to 70 years ago. And the carp were imported with Chinese pond mussel. Glaucidia insisted on their gills. They became established in the ponds. And we're making an extensive survey effort with them, doing a lot of environmental DNA work, extensive eradication work with Conservation Foundation. And they've been probably 90+% successful in kind of eradicating them. And there's plans to make one last strong effort at eradicating them next year. But we're still getting positive hits, in 2 kind of ponds in the impoundment, as well as in some spots downstream, and one anomalous kind of location on the Red River where we're not sure. We've surveyed it. We're going to survey it extensively next year. Was it a dispersal from those ponds in Hunterdon County that made it to the Raritan River, or was it a secondary accidental introduction at some point? We're not really sure, but I give a whole nother talk about Chinese pond mussel invasives on another day. The thing with that is the biggest thing that we're concerned about is why we're worried about this Chinese pond mussel is that in Europe, they're spreading rapidly. And it's kind of like the zebra mussel in lakes and ponds and other places in North America, where you go from a natural, green algal rich pond that is now crystal clear and you can see the bottom because there's so many zebra mussels and they're filtering everything out that they remove the bottom of the food chain. The same thing happens with Chinese pond mussels in Europe, and that they become they can get to extremely high densities in that they completely filter everything out of the stream. And you think how bad could it be to clean things better? But when you clean everything out, it's not good for the bottom of the food chain. And they outcompete the native mussels for food and forage, and then they outcompete them for space. So they're simultaneously gradually squeezing out a lot of Europe's mussels and causing a lot of other problems and spreading rapidly. So I always like to touch kind of in a little history and how we are and why things are where they are, and then we'll get into more of the details. One thing to think in your head is how and why are the mussels that we have here, and why are they where they are? And the changes over the last 20,000 years, kind of post-glacial, kind of drive what you see today. All of North Jersey was underneath of ice. So all the mussel populations, fish populations, everything else like that were kind of gradually established over time. So you had refugia kind of in the southeastern United States. And as the sea level began to rise again and the ice began to retreat, you had the gradual dispersal of fish northwards from there. And with them came the mussels. And over 20,000 years, they gradually marched their way northward. And what it does is it kind of created a big split between the Mississippi drainage and the Atlantic seaboard. So when we look at mussel conservation, there's a lot of stuff that's the Atlantic coastal shelf discussion and the Mississippi drainage discussion, with some closely related species that have a common ancestor. And in New Jersey, you kind of get that split with some species where you have brook floaters in both the Raritan drainage and the Delaware drainage that have been isolated from each other for a very, very long time, which kind of presents conservation questions in that they're the same species, but they've been geographically isolated for a very long time. So it's just a fun thing to kind of keep in the back of your head and think about how has this came to be? When we look at the substrates and the environments and what's there now, how did it come to be? There's a lot of interesting human use history with mussels. Native Americans use them as a food resource pretty extensively. You'll get shell middens and shell piles for these mussels where Native Americans use them as food. And it's funny, we used to always have our Peak West open house every year. And I'd always do a mussel display there and talk to people. And every year there were several people. There's always a bunch of people that asked if you could eat them. And there was always at least 1 or 2 that had actually eaten freshwater mussels on occasion. Usually floaters or other big things they found in ponds, and universally everybody said they kind of taste like nothing. It's a weird texture, but they don't have any flavor and it's kind of gross. So there you have it. Everybody always asks, can you eat them? Well, they can be eaten, but I don't know if you want to. The other interesting thing is there used to be a big button and pearl industry, particularly in the Mississippi, where these mussels were harvested extensively. Any shellfish will occasionally get pearls. So if you find a pearl, you're going to go out and find more mussels and try and find another one. And then they use them pretty extensively for making buttons. They would harvest them. They had special machines to drill them out. As you can see here, it's a drilling machine, puts a little punch holes in it. And there was a lot, just wide scale, like, wiping out mussel populations in many areas. It wasn't so big in the Northeast because we don't have as many mussels and wasn't extensive, but like some of the pearl industry, like it happened in, northern New Jersey, New England. And, people, once again, like if there's a chance of finding a pearl and a mussel, you and your friends go out and find as many mussels as you can and shuck them and hopefully find a pearl. And I do have a little shell button here that I found one time, which was kind of cool. My church's thrift shop had like one of those tins of buttons that everybody saves and has laying around. And I'm like, with this many buttons in a tin, there's got to be a shell button in it. And the interesting thing is you can see some of the ridging on the shell. I probably can't see it through the camera, but you can see some of the actual shell color and ridging on the other side of the button. So it's pretty wild. These things were like used by humans for a lot of different things over time. So there's a lot of things that, caused negative impacts to their populations. So that's some of the history and background. How do we got to where we are today? Why do we have the species that we have here? So how do we survey for them? They are incredibly challenging to survey for. They're very cryptic. They blend in very well. And, you're looking for actively siphoning mussels, but the moment the shell's closed up and a mussel's just resting and not siphoning, it blends in incredibly well. So it's a very challenging thing to work with. Also varies with your budget, depth of stream, lengths to cover. Like the size and depth of the stream can really make mussel surveying easy or hard really quickly. It is done through snorkeling gear, viewing buckets, aquascopes. If it's deep water, it has to be done with scuba. And you're looking for, exposed siphons, exposed mussels that are actually just sticking out, siphons, trails. Many mussels are completely under the substrate. So any kind of like large survey effort that we do, you might do several passes of transects to look for just obvious mussels. And then you might do like a small quadrant where you dig down in and look in, because especially the smaller, younger mussels are more likely to be buried in the substrate, because if it's a coarse enough material, they can filter just fine buried down a couple inches because there's good water flow. And you're small and fragile, so why not be buried and nice and safe down deeper in? So sometimes to find those other individuals, you got to like dig down in a little bit. And then, just to look more closely for inactive mussels or partially buried mussels. An interesting note, had a recent meeting where there was it was a reintroduction when they bred a bunch of mussels. Reintroduced them to the site. They all were tagged and extensively monitored. They had pit tags, everything else, the whole shebang. And they were doing it for like 13 years. And they put out, let's say 100 mussels numbered. And like in year 13, they saw I think it was 2 or 3 mussels that they hadn't seen since year 1. Like they just hadn't physically seen those individuals in those intervening 13 years. And that's with monitoring the site and the population. And all of a sudden, this year, the numbers 27 and 43 showed back up. So they must have been buried out of sight, just whatever, got missed for whatever reason, so they can be hard to find. You can see the trails on the bottom where they've moved along. They can move pretty far. If they get washed up, sometimes they'll move down in, back into the water. And it's just doing standard transects, doing time surveys. There's a lot of different ways of going about it from an organized point of view, but it's mostly just physically looking for mussels. And it's very much just a thing that requires a lot of practice and gaining a search image. These are just some photos of us doing transects and surveys in different places. The first one was in the Stony Brook, and then the second one in the Delaware River. And going out and just surveying for some more freshwater mussels. Snorkeling is done extensively as well. The aquascopes are really hard on your back over time. It's always a trade-off. When the water's really shallow, an aquascope's nice instead of being face flat in the mud. But when there's any kind of visibility challenges or turbidity, having snorkeling gear is nice because you can get up a little bit closer to the bottom and get a little bit closer look on things, So it's a trade-off. It's good to have different tools in your toolbox for how you go out and look for these things, because the same technique's not always going to work the best for every circumstance. And at the end of the day, hoping to find some of our native mussels and help track their distribution and help practice some conservation for them. That's a photo of a brook floater and a triangle floater that we found up on the Musconetcong one day. So getting into what we do at Endangered Nongame Species Program, and just checking messages, make sure somebody's have a question or comment. We are funded through state wildlife grants. We also get some outside grant funding to do survey works. We a lot of times we're doing time searches and runs, pools, riffles for qualitative. So mussel surveying, kind of goes 2 different speeds. And it's always a trade-off. Like you can either put all your time in a small area and not miss anything, or you can try and cover as much as ground as possible. So a lot of times we do time searches with transects and things like that, trying to get a good overview, cover a large area. And then if there's some kind of like really nice habitat or substrate or some area with a lot of mussels, we might come back and do a quadrax sampling or something more intensive to get a better count on them. Some of our current research is going on. There's a ton of regional interest in mussel research right now. There's a lot of different working groups, a lot of like federally led projects. There's a lot of species proposed for different listing statuses. There's a lot going on in the mussel world right now. And then like in New Jersey, there are an extensive amount of projects always going on. Like New Jersey is a densely populated state, so there's always something touching the water that involves mussels. At least that's been in my experience for the last 2 years with dealing with all the regulatory side of things, between roadways, bridges, bulkheads, piers, pilings, like you name it. Like it can be a couple of projects a week come in that might be having impacts on mussels or mussel habitat that require some kind of regulatory review. Environmental DNA is a relatively new thing that we're working on. We're looking to expand our capacity with it within fish and wildlife. Right now is mostly initially focused on Chinese pond mussel, and then looking to expand to using it for other rare species. Because these things are cryptic and hard to find, you can't always physically see them, you can use environmental DNA to detect them. So the idea being is you can take a sample of water, identify the DNA in it to the species, and see if that species is present or not in the environment or place you're looking. And the idea being with the Chinese pond mussel is we have a grid of ponds that we just like, pretty much nuke to try and kill the mussels in them. Are there still mussels there? And, so we used it for that. And then have they spread beyond there and things like that. So that's one of our newer tools. And we're starting to look into it to try and use it for identifying rare species in that, like, we've got a lot of historic records or old records. We may have gone back and resurveyed and not found any. Are they completely gone or did we just miss them? So that's where it's going to be a very useful tool for that. And then some of these species, like we can put a pretty good day survey effort in with a crew of 3 people and be very excited to find one brook floater. Like that's a good day. We found one, so it's like the odds of finding that or missing that are pretty high. There's a Delaware River Basin kind of mussel conservation kind of working group that's looking at a lot of habitat modeling and data sharing, kind of looking at the Delaware Basin as a whole and trying to look at mussel habitat within it from a mapping point of view, which is really kind of cool. We're also working on a Raritan River Basin project with the U.S. Fish and Wildlife, and they're using NRD funding, and they're looking at watershed level kind of like surveying inventory work and focusing on like any and all potential projects that may occur like in that kind of area. And it's really kind of interesting because so many times like mussel surveys and concerns are reactive in that, okay, we're going to remove this dam. We need to go survey mussels there because maybe we don't have data. We don't know what's going on there, and then deal with them, when we find what's there. The idea behind this is like, how cool would it be to just survey the whole river and see where all the mussel habitat is within that drainage, and then see what habitat is around any and all projects that may occur there. So that way we're kind of like a step ahead in the planning process. And then, okay, we know these projects do not have mussel concerns, and we know that this one has this concern and that one has that concern. We kind of have it as a planning tool for like, how to prioritize things and how to work through things. And it's also important in that if habitat is limiting for a species, like you might have a lot of mussels around a dam, but they may be evenly distributed up and down the river from there. Like there may be plenty of habitat for them. So moving them some short distance away is not a big deal. Versus you may have a project that's going on at a dam, and that's the only suitable habitat for that species for hundreds of meters to thousands of meters. So that presents a different challenge from a planning point of view. So this project kind of gives a snapshot from above. Dwarf wedge mussel, where it's a federally listed species, they're looking to update the status assessment in the near future. We do a lot of work with that. There's a yellow ant mussel working group focused on that species and is only found in the Delaware River in New Jersey. Historically occurred in the Raritan and Hudson. It's one of those species that's only found in one kind of drainage in New Jersey, and there's a lot of regional interest in other states. And an example for this from a conservation challenges point of view is that, with the introduced native species, sometimes you can get introduced native species that are closely related to your rare species. So in the the Potomac River, they have yellow lamp mussel, and I'm blanking out the name of the other species. I thought it was a pocketbook. I forget which one. Was accidentally introduced as another native, but it's closely related to the yellow lamp mussel, closely enough that they interbreed and hybridize. So now the yellow lamp mussel is being displaced by hybrids or the other species in the drainage. Brook Floater Working Group, we have a big grant doing a lot of work on this species, pretty much extensively surveying all the habitat and populations in New Jersey, which is really cool, and collecting genetic data range wide to compare the different populations to see how closely related they are. And here's a photo of us collecting genetic samples from a mussel with some of our U.S. Fish and Wildlife partners. And thinking of partnerships, we couldn't do a lot of this work without U.S. Fish and Wildlife, some of the other people that we're working together with. It's really great to have a bunch of good people to work together with. And they use like a little nasal speculums thing for like opening up your nose for a doctor to look at, but it's perfect for opening up a mussel to then carefully insert a Q-tip or swab to take a genetic sample. We've looked into like having a mussel hatchery in New Jersey. Like this is being done in a lot of other states. We've only getting some steps in that direction, but we're looking at some of the feasibility of it. Like, a lot of places, like the reproductive rates of mussels and the actual natural spread of them to, restored habitats is so incredibly slow that, like, we remove a dam and restore the habitat. It will take the next 100 years for mussels to get there naturally. So there's a lot of work going on with mussel hatcheries and rearing mussels to kind of expedite that process. And we've got a good potential to do it at Hackettstown. So we're kind of like looking into it, either that, you can rear mussels from like adult through host fish to a stockable mussel, or we can contract with another hatchery that does mussels and have them give us mussels that are, small, like intergrade size that we can then rear to a stockable size. So some interesting things. So getting into like dams and some things a little more interest, there's a lot of dam removals going on, a lot of interest in it. And it kind of cracks me up is like, there's a lot of dams coming out in the next couple of years in New Jersey, which seem like, facilitates connectivity for the host fish to be able to, relocate, move back through the environment, restores natural stream conditions. We run into challenges with like the lower head dams and the tumbling dams versus impoundments, in that like some of these places kind of subsidize and create habitat for mussels because the stream's been altered so much or channelized or blasted through because of high flow, that having some of these smaller features actually creates a break in the habitat sometimes. So it becomes like a conservation challenge when like you got a small dam you want to remove and it's like, there's a ton of mussels all around it, because they kind of replicate lost natural features like such as beaver dams and other things and log jams that used to be more commonplace in the waterways that don't exist anymore. The challenges with the geologic time scale. We remove these features and restore the stream. How long will it take the mussels to get back on their own? They may never get back on their own, without some help along the way, but it's a step in the process. And then we run into the concern with invasive species spread too, sometimes with dam removals, especially with like brown trout, snakeheads, and flatheads, in that like for native brook trout and some of our native fishes, like you might have some small dam feature that's keeping the invasive species out. So it's like there's certain dams we may want to keep just for that express purpose. They may not be good for other things, but it's like, well, we're keeping brown trout out of a brook trout stream. So just an example for everything. There's always some example contrary to what we think. I always look at dams like good, bad, and ugly, and we'll go through some like different photos, kind of descriptions of them and things like that. Dams vary in all kinds of shapes and sizes, so it's kind of hard to have a universal, take on dams related to mussels and other things like that. At the end of the day, like restoring connectivity and natural streams is like that's the best thing we can do, and like that's a net positive benefit. But it gets complex along the way with trying to like conserve some incredibly rare, species. With some of these mussels, like you may only have a couple individuals in like a couple kilometers of stream. So like, having a mortality event during like a dam removal or a bridge project or something else, like could have a significant impact on some of these species, especially if you have like a small isolated population, which happens quite often. So your dams vary in size, like, everybody knows that in this group. Columbia Lake being an example of a nice big like dam that's negative impact on a lot of habitat, versus like County Line, which was fairly recent, was a little more diverse kind of site with a lot of mussels all around it. So my little kind of toolbar kind of covers up some of the text on the slide, but I'm hoping you can see it. So sometimes with dams, they subsidize or create habitat for mussels, and it makes things complicated because sometimes they've kind of created habitat in some place where mussels may not have naturally occurred anyway, you know, like, and I've seen it in a couple instances, and here's a perfectly good example of that. For those of you who are familiar with it, this is Hackettstown and East Ave or whatever it is here, and there's a cemetery there, and Beatty's Mill Dam here. And what happens is the river kind of comes down, bypasses the old dam, and then keeps going. And this is a huge area of like not good mussel habitat, except for right here. And what happens is when the gradient, I hope you can see my mouse as I slide along here. So the gradient decreases and the flow rate decreases on approaching this bend. And what happens is you get a lot of sediment deposition on the upstream side of this feature here, that things like triangle floaters and brook floaters and elliptios like really like. So what happened was you have like a rather concentrated population of mussels on the upstream side of this old dam feature here that was created by the dam, you know, creating like this slowdown in flow and the deposition of sediment. So what's going to happen is when this dam removes and this becomes straightened out, like this is going to, all that sediment is going to flush and be transported probably downstream. And this probably, like mussels probably would never have occurred there if it weren't for the dam, because most of this is really unsuitable habitat. And when they were looking to do the relocation, like they had to move them pretty far away from here to find like someplace suitable for them to go to. So it's kind of an example of like a dam feature that's creating mussel habitat. So it's one of those things that make things a little complicated when we go to remove it, because it's like, oh, we gotta try not to harm these mussels. Another example was Burnt Mills on Lamington River, was like another really hot spot kind of thing where like the dam had created habitat for like mussels over time. So you gotta remember, like all these dams and features were there for like 150, 200 plus years. So all the wildlife doesn't really care why it's there. It's adapted accordingly in both good ways or bad ways. And if it happened to create some good habitat along the way, like you now have rare things sitting there around a feature you may want to remove. And then you have like your bad dams and things like that. You get big things like Columbia Lake, and this is not Columbia Lake. This is I'm blanking out the name, but it's on my slide. You have like larger dams and rivers that create large areas of impoundment and sedimentation. And what happens is now you have like these bad things like Columbia and I'm losing my voice. Island Farm, some of the other big ones on the rare things. And what happens is you create like a large area of unsuitable habitat upstream of it, you know. So you had the Musconecon River that was clear and flowing. Now you've created a big stop in it. It's a large barrier to any kind of fish movement. Like we've gone out and physically surveyed this. This is not good habitat for a long way upstream and not good habitat downstream of it either. So you have, you know, the large dams that are creating your biggest negative impact on mussels. And then I always put like ugly or complex dams. This is a perfectly good example. This is a county line one, and not to beat up on it, but it was like, this is a complex feature. Like it had several barriers, had several side channels and everything else like that. So we get some of these dams and features where it's like, there's mussels kind of all over in there, and they're not like, you know, they're kind of like evenly distributed all over in there. It's not like you got hot spots like that one in Hackettstown where it's like, oh, this created habitat here and there's no habitat in all these other places. Now we got to move them pretty far away. This one was like, well, it's not really hurting mussels. So it's like, what do we do with this now? You know, and it's easy enough to kind of like do a relocation to move mussels out of harm's way. But from a conservation point of view, it kind of like always, it brings up the question when we run into dams and features like this, where it's like, well, it's not really hurting anything. We leave it alone to remove it anyway. It's like, well, we remove it's not hurting anything either, you know? So it like makes it complex from a decision-making point of view, especially on the regulatory side of things, you know? Warren Glenn, thank you. I completely blanked out on that. I'm like, you have those moments where you're like thinking about too many things. Thank you. That photo was Warren Glenn. And then we'll answer questions towards the end, you know. So some of these, you know, become very complex things in that, like, you may have a complex feature, like county line being a good example, where you have different mussel species spread throughout the entire area of the dam. And then how do you deal with them when you go to remove it? You know, how do you relocate them? Especially if you had like different species that might require different substrates or different places to go to. So this is where, you know, some of these dam removals become very complicated, you know, from a conservation management point of view. And, you know, everybody from TNC can definitely speak to that, you know, as everyone's different than the next. Never know, my computer doesn't advance. So how does this tie into regulations and surveying and everything? becomes complex really quick. So there's INSCA, Endangered Nongame Species Conservation Act, prohibitions against take, in that you cannot take or cause mortality to listed species of wildlife. So you have to prove either you're not going to cause take or take steps to not have take occur. It gets really complex in that, like in aquatic and freshwater environments, we have a lot of protections of habitat, versus like in terrestrial environments, we can protect the animals but not the habitat, which is really a pain. And then there's like kind of like two parts of how it works on DEP side. You have the Bureau of Watershed Management that handles the regulatory side of things, like the law, the permits, and things like that. And the Endangered Nongame Species Program that handles the biology. And we work together hand in hand on things, especially when there's like species like mussels, where maybe the Bureau of Watershed Management doesn't have like in-house expertise or wants to have like a second opinion from us or something else on like some issue. So that's how we work together. And like when it comes to like permitting, like I just handle the mussel thing. Like it's like, like my little silo on tunnel vision. Like when it comes to permitting, you might have Andromeda's fish timing restrictions. You might have timing restrictions related to turtles. You might have other things, related to like flood hazard and all these other rules and regulations that come into play on these things. When it comes to New Jersey, we don't have a certified mussel surveyor list, and nor do we have an official mussel surveying guideline. The reason being, it's incredibly hard to get like things like this formally adopted by a government agency. Like it's just, we've tried, and it's just, there's a lot of bureaucracy in government sometimes. It's hard to get some of these things made. So it's never really like a clear-cut answer to some of these questions some days. And then with some of the other complexity, when it comes to like some of our data and landscape mapping becomes out of date because we're not able to get it, updated with newer occurrences and records in a reasonable amount of time. We can go into more detail about all those challenges offline. Some of the things we've done to kind of help deal with this problem is that U.S. Fish and Wildlife in West Virginia developed a standardized kind of guideline. It's called like the West Virginia Protocol for mussel surveying, and it was developed by U.S. Fish and Wildlife to be like a standardized thing for everybody to use nationally. So a lot of states are adapting it or referring to it directly. And one of the steps we've done in New Jersey is defer to those guidelines when it comes to mussel surveying requirements around a dam or some other kind of feature. And it's a very good document, and it gives you very clear kind of guidelines on what work and steps need to be taken in the process. U.S. Fish and Wildlife does have a certified dwarf wedge mussel surveyor list. So kind of like anybody on that list would fulfill the qualifications for being a mussel surveyor in New Jersey from a contracting point of view. We don't limit to that list though. So like anybody has adequate training and experience, like we will review like people's like resumes and CVs that like might be coming in on a proposed project to make sure they have adequate mussel experience. And I've seen it go both ways. I've seen where people have had like contractors that they're looking to use for surveying that really have like no field experience and maybe one class. And it's like, that's not really going to work. And then on the other hand, we've had people come in that like, I have no idea who they are, and then look through their CV and, oh, wow, this guy's done like all this work and research, all this other stuff. It's like, yeah, perfectly acceptable as a contractor. So it's common questions, about how we work with mussel surveys and mussel surveyors. Survey guidelines, between May 15th and September 30th, when water temperatures are above 55 degrees Fahrenheit. Reason for that is twofold, in that water temperatures below that cause higher mortality and stress to the mussels, because once it starts to get cold, they're looking to bury down the substrate and be inactive for the winter. So you get higher mortality when trying to remove or handle mussels in colder temperatures. And then on the flip side, like they're just less active, less visible. You're more likely to just miss them surveying because they're not open and not siphoning, or they're buried into the substrate. So I know this becomes a limiting factor for a lot of people. It's like you can't really survey for them during the winter because you can't see them. And if you do see them and try to handle them, you're going to cause mortality. Stream conditions and clarity need to be suitable for surveying. It's always a challenge with stream flows and turbidity and visibility. We always do relocations need to be done by like September 15th. Just because we have a 2-week and 1 year post relocation monitoring requirement. So like September 15th, you need 2 weeks till September 30th to be able to do your follow-up check, So a lot of times people will like be looking to do relocations, like try not to put it up against the end of September, because like if you have poor conditions or a tropical storm comes through, next thing you know, you're not surveying, And we do allow some flexibility, because the other thing with climate change and global warming, like conditions are sometimes good well into October to still survey for mussels sometimes, but it's not predictable, you know. For us, you know, when it comes to project management, you know, submit a mussel surveying plan for review. So let's say you got a project, it's got a hit for some rare species of mussel, you need to do a survey. You know, contract surveyors put together a surveying plan they've reviewed based on the West Virginia guidelines. You know, we will review it, make sure it's acceptable and sufficient for what we're looking to see. And for the most part, like everybody does a really good job. Every now and then we sometimes have to tweak things, you know, and stuff like that. You're looking at an area of direct impact from the project and any appropriate buffers around it. When it comes to dams, like you're looking at the area of direct impact being like the dam, the area where you're working around the dam that's going to be immediately impacted by the change of flow, as well as the upstream impoundment where like the water level is going to change or drop. So the idea being is you're going to survey the area around that feature to look at what's going to be directly impacted, plus like a buffer upstream and downstream of it, you know, 100 meters downstream, 50 meters upstream, generally speaking, to kind of get a snapshot of what the area is, what's going to be impacted, and what is found in there, you know. You can do it with, we'll do it with transects and different grid cells, collecting size and substrate details. And so much of this is driven by the size of the stream in the project. You know, like small little streams that are narrow are like easily surveyed versus you might have something on the Cooper River where like you have a big chunk of stream bottom that has to be divided up. And sometimes they'll do randomized survey plots within it. Like, you know, there's some flexibility with surveying. Like, you know, we do use like a certain amount of common sense when it comes to these things. Like we're not going to have people, you know, survey hundreds and hundreds of square meters of stream bottom unnecessarily if we can avoid it, you know. If there's freshwater mussels and habitat present, they develop an impact avoidance plan with like dealing with them, you know. So you go out and survey your feature, you find some brook floaters there. Now what do we do? You know, so some steps in the process could be like a cancel or redesign. Generally speaking, with like large projects, like if you're replacing a bridge, a road, or moving a dam, like you're not canceling that. Like the bridge has to be fixed. So the mussels have to be moved. You know, with some stream enhancement projects, what we've done is just tweak the design. Like, you know, like, oh, great, we've got this one beautiful mussel bed here. You know, we'll just, you know, move the pool you were going to build over there. Like, no big deal. You know, sometimes I've done this too on projects where just changing stream access points for equipment. Because a lot of times it's running heavy equipment. And I remember having another project where it's like the only place that was going to be impacted was where they're running the equipment into the stream. It's like, well, can we get it in the stream some other place? Yes. Okay, cool. Problem solved. Sometimes just silt runoff control, you know, these things are very sensitive to, like, you know, they can tolerate a certain amount of silt and runoff, but they're not designed as an organism to be completely clogged and choked by silt and things like that, you know. So sometimes it's just very basic controls in that, like maybe you've got a project where you're doing a road and there's no direct impacts, but you're going to generate a good bit of silt or runoff. It's like, well, take steps to mitigate that, and their concerns are addressed. And then last thing is relocation salvage, moving mussels out of harm's way. And that involves, you know, going through the site, doing repeat survey efforts, trying to find and collect every mussel that's liable to be, you know, negatively impacted by the project, and moving it to some area of suitable habitat out of harm's way. You know, we always, you know, have to approve all the relocation sites to make sure it's suitable habitat for the species. Usually trying to be more than 150 meters away. Just because, like, and usually try not to be downstream of it, because like if you're doing something to impact the stream, you don't want to just move the mussels a short distance out of the way downstream because they're still going to be impacted by it, you know. But then other places like the common sense comes into play. Like you get like in the Delaware River or like South Jersey where there's just mussel habitat everywhere. It's like, well, we can, you know, move it some reasonable distance closer than 150 meters if it just makes sense, you know. And then with 2-week and 1 year follow-up monitoring to look at survival and, you know, see, you know, hopefully, you know, still be able to find the mussels again. You know, they can be tagged with like alphanumeric or color tags. Sometimes you use pit tags. You can use a pit tag scanner for them. It's kind of a trade-off for using the pit tags because they're a little bigger, bulkier on the mussel, so they can cause some problems with the mussel. But it is like if something's buried in a substrate, it allows you to find it again easier. You know, so sometimes with rare species, we might, you know, if you got 100 of some rare species, we might require some subset to be pit tagged and then the rest just be numbered, you know. It's always tough talking into a computer screen here. So like permitting, getting into that was always a good point to bring across to people, is like our mussel surveying permits is like a DEP scientific collecting permit. It can be issued independently of other DEP permits. Like the mussel surveying permits is ran by us in Nongame. So like if you're interested in practicing conservation for mussels on some stream that is on your property and you want to identify what mussels are there, I can issue a permit to do that. So there's a scientific value of what you're doing. We can issue that permit at any time. Same thing can be done for any dams, features, or other projects. Like if people are looking to be preemptive, it's like we can give you the permit to survey at any time, and then you'll know what you face related to your project. So the thing I've ran into a lot of projects is like they'll be waiting for all their other DEP permits and everything else to be in play. And now all of a sudden, like, because everything takes longer and delayed and pushed off, all of a sudden it's like September and they're like, oh, we finally got all our DEP permits. Can we do our mussel surveys now? And it's like, oh, no, not possible. Now you got to wait till next year. And the idea being is like, well, if you need to do a mussel survey, like we can get that going anywhere during this process, and then you kind of have like your ducks in a row, and then we can deal with the other problems later, and then there's a whole other caveat when it comes to permitting. If there's federally listed species and coming to the federal dwarf wedge mussel, surveyors list and U.S. Fish and Wildlife, is that like if you have federally listed species and there's federally listed species there, you need to have a federally approved person to do the surveys and handling on that list. So like we've had projects where like somebody's like surveying for like state listed species, also you stumble on a federally listed species, like it changes the whole ball game from a permitting point of view. They need to do permit coordination and consult with U.S. Fish and Wildlife. So I speak more on just the DEP side of things, and then if it escalates to the federal side of the thing, is we loop in all of our federal partners as well, which recently happened on a bridge project up in North Jersey. So most of the rest of the permits are independent of scientific collecting permit. And one thing I always try with people is like, if we can get the sooner you get the mussel surveys done, the better off you are, because then you know what you're dealing with, you can plan accordingly. Because so much can't happen until we know whether there's mussels there or not. And sometimes you go there and survey, and there's no habitat, no impact, no worries, and takes a lot of stress off people's plates. Here's just kind of a map and an example of like what like a mussel survey would look like. You have an area of direct impact around whatever feature you're looking at, like the red line. You could imagine being a dam or a bridge or something else. And then you have an immediate buffer area around it that's the area of direct impact, where equipment's going to be ran, where the stream flow is going to change significantly. Maybe there's some little pond there that's going to be now drained. So that's the area that's going to be directly impacted by what you are doing. And then you have some survey buffers upstream and downstream of it, 100 meters, 50 meters. And when you go to the guidelines in like the West Virginia guideline, the U.S. Fish and Wildlife and the West Virginia guideline, like these buffers can vary depending on what you're doing. So if you're doing a bulkhead along a shoreline, you might be looking at a 10 or 20 meter buffer along the shoreline where the bulkhead's going in, because the impacts are going to be relatively localized. Like you're moving a dam, like if you're doing dredging and creating a huge amount of silt and other stuff, like it might be a lot larger buffer. Within that area, designing transect surveys, doing grid cells and other things like that based on the side of the stream and how much square footage of stream bottom do you have to deal with. Here's just another example of like a grid cell layout from that kind of guide, in that you're looking at the areas being directly impacted with a more focused, intensive survey effort. Maybe even doing smaller cells in a really focused area close to it. A lot of this is driven by what you're seeing as well. Like if you're going in there and you have really dense mussel beds and really good habitat, like you're going to adjust your kind of grid cells accordingly. Or like you're going to say, hey, like this is a huge mussel bed, there's mussels everywhere, we're just going to do like kind of a random quadrant sampling to kind of get a snapshot of what's going on here. And a lot of times like the squiggly lines around it is kind of just doing a quick kind of qualitative survey around whatever feature you are, whether it be like random searches or transects. The idea being is like you have your direct impact area here and some of the buffers around whatever you're doing, but then getting a snapshot of what's going on around your site. And that comes really important, especially when you got to go back and look for like a relocation site. So like all of a sudden, like we need to move mussels from here. It saves you a lot of time and money to have already done some kind of like preliminary surveying in the area to find like a relocation area instead of having to go back again. And, you get a feature like this where it's like, this is all mussel habitat and all good mussel beds. It tells us like, okay, relocating these mussels from here to here is a pretty straightforward thing. We go survey this and you got a mussel hotspot for whatever reason right here, and then like a wasteland around it and no mussels. It's like, well, we don't want to relocate mussels from here to here because it's not good habitat. We're now going to need to look farther away or look for, suitable habitat elsewhere. So I'm hoping that makes sense, and we can always kind of get into Q&A, questions at the end here. I'll quick go through, and I don't have a time. So how am I doing on time here?
Hey, Robert, this is this is excellent. I didn't want to stop you short. We are a little past the hour.
Okay,
but
that's
if
what I
you
was
still
worried
have
about.
a few more slides, that's okay. We do want to save a little time for Q&A, but if you want to wrap
This
up
was
your...
kind of my natural stopping point. So,
Oh,
which
okay.
is good, you know. I figure I'd
Good.
check on in. So
Yeah.
so after that, so that's just kind of like that's a lot of information to like dump out in a straight stream, and doing it over computers is tough. Like, you know, we can get together in person and do more of a like, you know, species specific thing and open up to anybody who's interested in coming out for it. You know, the next thing after this would be like the next part of this presentation is just going into the species we have in New Jersey, what they look like, and identifying them, which that can be for a whole nother day's discussion. But just, you know, some of the take home for that is like every one of them has a different habitat, different requirements. You know, like I kind of touched on it quickly as we went through this, but like, you know, your tidewater mug and eastern pond mussel are tidal freshwater mussels of southern New Jersey, not found in northern New Jersey. And then your other rare species are found in only certain streams, drainages, or features within those drainages in North Jersey. So you can have a certain stretch of river with little microsites of habitat where different mussel species are found. And then one last point to kind of get into, which is like kind of like one of those like newer kind of scientific, not newer, but it's like one of those questions we're trying to answer or understand better, is when you get multi-species assemblages, how important is that and what difference does that make? In that sometimes you will get extensive beds of like, especially like in the lower Delaware, you might have like thousands of alewife floaters with sprinkling of tidewater mug and eastern pond mussel mixed in. And the same thing will occur up in northern New Jersey, where you might have nice beds of large beds of hundreds of eastern elliptio with a sprinkling of you know, brook floaters or triangle floaters mixed in. And sometimes like these multiple species assemblages are an important part of the habitat for the mussel. So it's like you don't want to have the tunnel vision of like we only want to focus on the rare ones. Like some of these beds are important features for like all the mussels found there, and there's some dynamics going on that we may not know or understand. So with that, we can open up to Q&A and whatever people want to talk about.
Thanks, Robert. I see a question in the chat from Walt. I'm not sure if he's still on, so I'll go ahead and read it. It's with regards to some data. So do you know the numbers of how many of New Jersey waters are reliant on mussels for cleaning and filtering versus how many were historically reliant, and what the current goal is?
We don't, but that is a number we could get. That could be mapped and kind of extrapolated. Like the best example I could give though is like you have the Hudson, Hackensack, and Passaic drainages that mussels have been completely extirpated from. That historically used to have like, you know, eastern pearl shell, you know, all these places where used to have salmon runs and other things and sturgeon and all these species that are like now gone from those places, you know. So yeah, we could kind of extrapolate that, especially with some of the data we have. And there's a lot of ongoing surveys going on right now, which is like kind of exciting in that like we are surveying a lot of mussel habitat like all over. Like so in the next year or two, we're gonna have a really interesting snapshot of like mussel habitat in a lot of northern New Jersey, which is kind of cool, you know.
Yeah, Beth, do you want to go ahead with your question?
Sure. I've heard you mention several times that you guys are working on surveys in different parts of the state. So I was wondering if you were open to, you know, can anyone give you a heads up? And, you know, if it's of interest to you, can you help with surveying either by, you know, tag teaming, you do the pre-survey, somebody else can do a post. You know, is there a way? We see some of that with New Jersey Fish and Wildlife, who come out and do some fish surveys with relation to dam removals, and also the Bureau of Freshwater Biological Monitoring has, you know, lent their team to some dam removals, specifically with eDNA, but also with other types of monitoring.
Yes, and better chances of that now than there were even like a year ago, just because we have more funding and more surveying going on. Like, so like some of its limits, like the Raritan example, like that is the funding is limited to the Lamington, North Branch, and Stony Brook. South Branch was excluded. I forget why, but we're going to survey that with other things. But like it was kind of like a broad thing to look at features within that. So it's kind of like doing exactly like as you say, is like the whole idea of Project Bain is like we know. I think I forget. It's like 21 features in that area or something that like whether they be like dams, bridges, fill in the blanks where like work may occur. The idea of Bain is like we're going to do like kind of like a pretty extensive survey of that whole area with focuses on those features. So that can be something that we can look to do more of in the future, especially when we get past like some other survey priorities, when we start working the secondary sites. So like we're working through a bunch of surveys now through our prioritization, like tier one, like the things we need to get done. But I'm fairly certain that we're going to have like, fundage left for like quite a bit of additional surveys in the future. So feel free to reach out and we can talk about that, because a lot of that's changed like in the last year. Like when I thought about all the mussel work going on, like I don't know, like people have been kind of like, throwing money at us for mussel stuff like in the past year. So there's a lot of work kind of going on at different pieces of it, so it's kind of cool.
I have a question. Oh, go ahead.
I was just going to say that's great to hear. That's great to hear.
I have a question. So with all the data the new data that's being collected on mussels, does that data eventually go with or talk to the fish data in any way? Because you mentioned that so we need the host fish to help with the reproduction. So if you're making like those bigger assessments, is that something that you're also doing with the fisheries biologists?
Yes, that's definitely something we're doing and looking to do like next step in the very near future. Yeah, right now it's one of the things we're talking about, like Alex who's on there and like some other staff is like, so the problem with like mussel surveying over time has always been piecemeal, project related, or like it's like it was always Jeanette and then now it's just me and then I got Alex to help me out. So it's like it's very much like very limited to what we could get done. But like now that we're getting like some like, we just surveyed the whole Stony Brook this year. Like we surveyed most of the Musconetcong this year. Like we're doing big enough chunks at the same time that you can kind of get a meaningful snapshot that then can be compared to other things. Because like you look at our landscape data and some of our currents data, like we could have records from 1980 and 2023 in that map. Like, that's not really helpful. When we have a lot of data points and survey information, that's all 2022, 2023, like that's going to be really interesting to look. And like one of the things we're looking at is, comparing it to the fish data fisheries has, and then looking into the environmental DNA thing too, is looking at some of the stuff that like, Alex is like kind of an environmental DNA kind of guy, is like looking at how can we use that to look at the assemblages of species found in different locations and compare them, and how does that look on a map? Like, so yes, to answer that question, and yes, because it's, the next logical step. And like, we actually have the data and capacity to actually make it happen for like the first time, like probably ever, to be able to, have some of this kind of stuff occur, which is cool.
That's great. And one of the other questions in the chat, somewhat related, is are these mussel surveys of the Delaware River, is the question in the chat, below Trenton available, but more generally also, is this data going to be available online or through other portals?
It will be. We're in the process of updating all of our landscape maps. So landscape mapping is like how data is modeled out, So when that map is updated, like any new mussel occurrences will be going into that. So how that kind of works is like a mussel is a point on the map, but like mussels are poor examples. They don't move very far. So let's say a fish. So you got like a rare fish as a point on the map. Well, that fish is not like bolted to the bottom of the stream, like it moves around. So how we deal with that data is like that point now values X number of stream in some reasonable distance that the fish may move. Works better for butterflies than that. We know a butterfly will fly X distance, so we'll value all suitable habitat within X distance of it because that butterfly can move that distance. Same thing goes for like the mussel data and things like that. So like that data will then be viewable in that map, and that streams and drainages will light up for the different species that are found there. Part of the reason we do that too is data security. Not that people go out and collect mussels and sell them online, but like for other rare species where people do collect them, we try to like melt it back into the landscape in some natural, logical way, and that like you can't just tell where some rare thing happens to occur. But then for like scientific purposes and research partners, like we can do data sharing agreements and share like more specific point data for like some other purpose. So if there's need or interest in that, you just shoot us an email. We'll discuss the project. Like I partner with anybody and everybody that's interested in doing like conservation work. And it's just a pretty simple, straightforward thing to share point data for scientific purposes,
Great, thanks. And just, I'm not sure if you're seeing, but we did pop your email in the chat. So if folks did want to follow up about getting any of that data, feel free to do so. There's a couple more questions in the chat here. So one is, does the EPA Superfund project have to conduct freshwater mussel surveys for DEP prior to remedial dredging and or capping projects?
I don't know how to answer that one, like a specific project.
I think it's specific to Superfund projects.
So it all depends on proximity to documented habitat. So like, yeah, like if it's like next to a stream or river and that river is valued for mussel habitat, then yes, they would have to do like a survey or other work related to it, you know. So that kind of comes back to that mapping thing, and that like the landscape map maps all the mussel data based on streams and rivers. So if you have a project here and then, you know, let's say this is a stream valued for brook floater and your project is here and it intersects that stream, like now you trip like a regulatory red flag, you know, like now it's going through a more extensive review process in that, like, okay, you got to look into mussels, you got to do X, Y, and Z, versus like, here's your stream that's valued for brook floater and your project's here, it does not intersect, you know. And there's some caveats to that, in that like the landscape map is out of date and we have, you know, like I could get data tomorrow that won't be in that map, you know. So it's like there's always like, it's kind of like a screening tool in that, like it gets you close, you know, and it's not a perfect system. So sometimes the modeling doesn't work too well because, you know, streams and rivers are all interconnected. So you have to kind of break them up somehow. So it's like, you know, you don't want a mussel that's all the way up in like, I don't know, Clove Brook valuing the Delaware River all the way down to like Trenton, you know, so you got to find some logical way to break that up. And sometimes it's arbitrary, you know, so it's just the way it goes.
Thanks, Robert. I see one more in the chat here with regards to flood mitigation projects involving living shorelines to control erosion or replacing culverts. Are there ways to incorporate mussel habitat into these types of projects?
So living shorelines and yeah, a lot of people are looking at like PDA, Partnership Delaware Estuary, you know, Krieger's group and all of them are looking into that in different ways, shapes, and forms. It's kind of like a new thing for incorporating mussels into it and how they'd be used. Like we haven't really dabbled in it too much, and like there's always concerns, you know, both ways, because A, they can be used to restore habitat back the way it used to be and help clean water. B, but if it's not a good place for mussels to begin with, you don't want to kind of put them there to their doom, you know. So it's kind of like a trade-off. But they're doing a lot of work on that, more so now. And like, you know, we're kind of open into like the research and like science, you know, behind it. Like, you know, how can we restore these things, make it suitable and things like that. And then like the point we always kind of push to it as like monitoring before management, in that like, you know, if you have an area you want to, so many times people are like, I want to do this project here and make it better, but then there's no real understanding of what's there before. So it's like, we always like, you know, get a good survey effort, see what's there now. If there's not mussels there now, well, why is that? You know, if there's some kind of like abiotic factor or something else is just making it not good for mussels, you know, we try and answer that question. But like, if it's, you know, the habitat's been restored, the water quality is restored, and the mussels just aren't there because it's going to take the next 100 years for them to naturally disperse there on their own, then you can kind of look at like, you know, incorporating that into it. There's quite a few projects. There's none in really New Jersey. I think there's going to be one going on, or I can't remember if they're doing it or it's going to be doing it, because some of these things kind of come up and then go away. But I know in Pennsylvania, they've got a couple projects where they've got ongoing research. It's like the more these projects they occur, the more like science and information we learn, and the better we're able to do it in the future, you know? So it's like one of those things that's kind of like, it's really new, tip of the iceberg, and like, you know, we're kind of learning along the way. And you know, so many times people are so risk averse to these kind of things, and that like some of these things in these species are gonna go extinct, like no matter what. Like so we, you know, have to take some actions and try, you know, rather go down trying than sitting back, you know, worrying and wasting, you know, time away.
Thanks, Robert. So I'm not seeing any other questions in the chat. If others wanted to come off mute, raise your hand. We can stay on for another minute or so. I want to just say thank you so much to Robert for joining us today and for dedicating some time to this topic. I think there was a lot of interest. We had a lot of people on the call today. This recording will go on to the njdams.org website under the resources tab, and then also on the YouTube channel. We have one more info session that's coming up to close out this year, and that's going to be on Tuesday, December 7th at 1 p.m. and it's going to be on developing an RFP. And so more information will be posted on njdams.org. You will get a registration link there. And if you have any other questions or looking for more info, we're always around. Feel free to reach out. Thank you so much, Robert. We really appreciate your time.
Oh, thank you. Thanks for having me, and hope everybody enjoyed it, learned something. You know, it's a lot to kind of cram in, but
yeah.
And we'll look to like, I'd like to offer another workshop for like hands-on and going over stuff for people. So if there's people interested in learning more, learning ID, we'll try and do something over winter sometime. We're going to advertise it like this kind of group or something else, you know?
Okay, that sounds great. We will follow up with you. Thank you. Thanks all. Enjoy the rest of your day.